Xenobiology, a subdivision of synthetic biology, delves into the realm of crafting and controlling biological mechanisms and structures. Named after the Greek term xenos, connoting stranger or alien, this field diverges from conventional biology by exploring unique biological systems and biochemistries not currently present in nature. Departing from the established DNAR-Na20 amino acid framework, xenobiology ventures into uncharted territory by investigating xenonucleic acids as information conveyors rather than DNA or RNA. Moreover, it examines an expanded genetic code and the integration of non-standard amino acids, known as xenoamino acids, into proteins. In comparison to exobiology and astrobiology, which focus on detecting life in the universe, xenobiology distinguishes itself by attempting to engineer life forms with alternative biochemistries and genetic codes that differ from those found on Earth. This approach holds promising prospects for unraveling fundamental biological insights and shedding light on life's origins. By exploring alternative biochemical environments, researchers aim to grasp the underlying principles that led to life's emergence. Furthermore, xenobiology offers avenues for enhancing industrial production processes by manipulating biopolymers and bolstering pathogen resistance. For instance, initiatives like the Metacode project endeavor to introduce novel catalytic functions into bacterial cells, expanding the capabilities of proteins for more efficient material functions. A key feature of xenobiology lies in the concept of semantic containment, where genetically modified cells are engineered to resist virus or bacteriophage contamination, thereby enhancing biocontainment strategies. Innovations like genetic firewalls represent a crucial biosecurity measure, aiming to prevent horizontal gene transfer and mitigate potential risks to human health posed by traditional genetic engineering practices. By designing alternative genetic codes and biochemistries, xenobiology presents a unique opportunity to create biological systems that are incompatible with natural genetic frameworks, effectively sidestepping issues of gene transfer and fostering the development of novel synthetic auxotrophies. While existing cells incorporate only a couple of such features, research into alternative DNA forms initially centered on why RNA and DNA were chosen over other nucleic acids during early evolution. Experimental studies diversifying nucleic acid structures have produced innovative biopolymers like HNA, TNA, GNA, and CENA. The successful integration of XNA into plasmids and its use in DNA synthesis highlight the progress made in this domain. XNA's potential as genetic catalysts has been explored, with FANA and other XNAs showing promise as enzymes in cleaving RNA and ligating nucleic acid substrates during cellular processes. Efforts to expand the genetic alphabet involve replacing standard DNA bases with unnatural pairs. Various studies have explored new base pairs that function in transcription, translation, replication, and different forms of genetic synthesis. Additionally, researchers have incorporated artificial nucleotides into bacterial DNA, facilitating bacterial growth without developing mRNA or proteins, utilizing these artificial nucleotides. The challenge lies in developing novel polymerases capable of replicating these alternative genetic constructs. One method involves altering codon tRNA synthetase pairs to integrate non-canonical amino acids into proteins. Significant advancements include replacing all stop codons in the E. coli genome and reprogramming essential genes, demonstrating the capacity for massive codon substitutions. Moreover, altering the genetic code by introducing quadruplet and quintuplet codons has been explored in cell-free systems and bacteria. Directed evolution presents an alternative approach to substituting DNA with XNA, as shown by engineering E. coli strains reliant on synthetic nucleotides for growth. This shift in environmental engineering offers a unique pathway towards achieving xenobiological goals. Xenobiological systems are engineered to differentiate themselves from natural, biological systems, creating a barrier to genetic interaction. This segregation is highlighted through the concept of a genetic firewall, which acts as a safety measure by altering the genetic machinery of the cell and leading to what is called semantic containment. The idea behind the genetic firewall is to prevent information exchange with natural cells, thereby overcoming previous safety system limitations. An example of this theoretical concept was demonstrated in 2013 through the creation of a genomically recoded organism, GRO, that exhibited increased resistance to T7 bacteriophage. The potential to manipulate a large number of genetic triplets opens up the possibility of developing strains that incorporate XNA, novel base pairs, new genetic codes, and more, ultimately creating organisms that are completely isolated from the natural biological realm. However, any alterations leading to semantic containment in new organisms would still warrant thorough toxicological screening due to the potential for novel biochemical systems to introduce toxins or allergens. Various groups, including policy advisors in the U.S., European National Biosafety Boards, the European Molecular Biology Organization, and the European Commission's Scientific Committee on Emerging and Newly Identified Health Risks, SCENIHR, have recognized xenobiology as an emerging governance issue, with efforts to define and assess the risks associated with synthetic biology and research priorities in this domain.